All right. Well, thank you for the opportunity to be here. This is a little different crowd than I normally hang out with, and I was advised that to give a more presentation focused more on giving an overview of Tox 21, because my understanding is that this audience is not necessarily familiar with what's going on in the toxicology arena with regards to trying to implement some of these newer technologies within toxicology. So it's going to be more of a less, my group does a number of related Tox 21 research activities, so it's going to be less of a research talk and predominantly focused on giving you a background on what these activities are, and then I'll throw in more at the end. Two pieces of data that we've kind of pulled together from some of our studies that gives you a context of what potentially you could expect from some of these newer technologies, some of the limitations, and we'll have a discussion on some of those limitations, and then we'll get into more of what Joe was talking about how another study that we've implemented was trying to incorporate some of these nutrients as well as food components into our Tox 21 studies in order to provide a proper context to how we're interpreting results from chemicals. In that regard, this is hopefully what I believe, at least my understanding of what one of the things that you find interesting regarding this Tox 21 and may be more of interest to your particular research or efforts. So for those that may not be familiar with what's going on in toxicology, there's actually quite a broad movement within toxicology, multinational movement within toxicology, trying to redefine where we're going as a field and as a science. And this is due to multiple efforts both in the US as well as in Europe. You can see this is just a handful of handful of screenshots or logos from multiple different regulatory agencies as well as academic research programs, framework programs within the EU, all trying to basically incorporate new technologies, primarily in vitro, using cell-based or high-throughput screening or a number of other in vitro type efforts to try to screen many of these chemicals for biological activity. And you can see in Europe there's a couple of them. Let's see here, we'll get just the sensitive, trying to use in vitro efforts to try to predict skin sensitization, inflammatory type responses. There's another one, an in vitro effort trying to predict acute dose LD50 type values, so you don't have to run necessarily an acute toxicity study anymore. And ReProtect is another European initiative trying to focus on trying to use in vitro efforts to try to predict reproductive toxicity. So there's a number of things in Europe. And then in the US, there's a number of efforts like Tox 21. I'm going to give you a little bit more of an overview on what that's going on. Toxcast, which is an effort. It's also incorporated within Tox 21, but it's actually a separate effort within EPA trying to use these in vitro efforts to, in a toxicology-related context. All right? And this all kind of, it started before this particular report, but this report actually generated a significant momentum within the toxicology community, more as a discussion about where we're going as a field. And this all stemmed from a National Academy report in 2007 that listed at least one vision for how we should transform toxicology testing going forward. And this, coming out of the Academy, obviously carried some weight and actually was also good timing within the toxicology community because of existing frustration with trying to rely on these expensive as well as resource-intensive, animal-intensive in vivo studies and how we evaluate chemicals. And I'll give you in the next slide a reason, show you a figure of why that frustration had been building for some time. But nonetheless, the vision defined a transformative redefinition of toxicity testing in order to take advantage of modern biology. And this modern biology would then be able to provide a broader coverage of chemicals. With our current efforts in vivo studies, we're really confined with a number of chemicals we can really evaluate from a resource perspective, as well as be able to evaluate things like mixtures, which has been largely ignored in a toxicology community. And also be able to evaluate a broader range of endpoints and life stages. All the while trying to reduce cost as well as the time to be able to come to some assessment of the potential health risks of these chemicals. and also provide a more robust basis for risk assessment by understanding the mechanisms of how these chemicals act, and then be able to make a better assessment of what potential processes or potential hazards will exist across species, trying to evaluate them in rodents. Not all of that translates necessarily to being a risk in humans, and if you understand a better understanding of the mechanisms of how these chemicals work, you'll be able to make a better assessment of how that may translate into humans as well. And so, according to this report, that in the not so distant future, this toxicity testing would become routine and used predominantly in vitro models and trying to look at perturbations of what, at least in the report, called toxicity pathways. And we'll get into that a little bit more later. The reasons, as I mentioned, for underlying the effort to modernize toxicology and essentially try to, and one of the reasons for frustrations within the toxicology community is kind of shown in these two bits of data. On the top is essentially a timeline of when the majority of these current in vivo tests, the way we currently test chemicals, and when they were developed throughout history, right? They weren't necessarily, some guy didn't necessarily come up with a toxicity testing type strategy early on how we're going to evaluate these chemicals for safety. This evolved over time as we began to evaluate and began to use more chemicals in our economy, began to use them in different ways in the lifestyles and the products we use. And so these were kind of added on throughout the course of history. And you can see the number of these in vivo studies and when they were developed and for different purposes were developed decades ago, and not necessarily with the thought of being able to incorporate much of this new technology and an increase in biological understanding that's really come about in the last decade or so. The other part of the motivation for trying to transform toxicity testing is seen in the bottom graphs. And it's really the fact that we haven't done a very good job of keeping up with evaluating the safety of many of the products we're actually exposed to, at least in the broader environmental industrial chemical arena. The food and pesticide type arena is a different arena where there's regulatory mandates for evaluating safety of products. But in the larger sphere of chemicals, we really haven't done a good job, and this is kind of outlined below. And you can see on the left-hand graph, bar graph, is a number of chemicals in different classes. IRIS is just in INSEA, it's a class of chemicals which there's actually formal risk assessments done on those products. You have pesticides, you have candidate contaminant lists, predominantly in things like water that are mandated to be tested, and you can see the number of chemicals in those particular lists. Medium production volume chemicals, toxic release inventory, so that's more released in the air, inerts, and as well as high production volume chemicals. And so you can see the number of the chemicals number in the thousands in those different lists, right? And on the right is a percentage of those chemicals that have different forms of safety evaluation, different animal studies associated with them. You can see the majority, close to 60%, have an acute tox evaluation, but it drops off significantly after that. The percentage of chemicals with genotoxic, evaluated for genotoxicity, number of chemicals evaluated for reproductive, cancer, and developmental toxicity are all under 30%. So we really haven't done a good job for many of these chemicals for broadly evaluating the potential effects of these chemicals on human health. So this is kind of a motivation for why these newer methods are actually need to be implemented in order to increase our ability to evaluate both these legacy chemicals as well as the number of chemicals that are being introduced on a yearly basis. So to address this challenge, a couple of federal agencies, predominantly the EPA and the NTP through the Tox 21 initiative, although in addition the FDA has also joined the Tox 21 initiative, formulated a new plan to try to incorporate some of these new technologies, predominantly high throughput in vitro screening, in order to evaluate these particular chemicals. And the Tox 21 began in 2005 with the National Toxicology Program and NIH, National Center for Chemical Genomics, as well as in partnership with the EPA, joined in 2006, and they signed a 5-year MOU in order to be able to perform these high throughput in vitro assays, predominantly at the NCGC. All right, the US FDA joined this consortium in 2010 as kind of a multi-agency effort to try to use these new technologies and new efforts. On the right-hand side is an effort that's predominantly being led by the EPA, and this was initiated inside a newly formed center within the ORD of the EPA, and it's the National Center for Computational Toxicology, and it began in 2007 as an effort that spurred within the organization. And this is actually quite different from a federal agency perspective. In the past for the EPA, it wasn't necessarily generating a lot of data on a particular set of chemicals, right? That wasn't necessarily coming from the government. It was being driven from industry, generating data on their products. Well, this is a change in that initiative where the government was actually generating a significant amount of data on different chemicals that were in commerce. So this is kind of a switch between who's generating the data and who's interpreting that data from what the previous paradigm actually was. And this was set up to be in two different phases. Plan was phase one, which was supposed to complete in 2009, where I'll show you in the next slide the number of chemicals and the plan for that particular set of phase in terms of what was being assessed. And then a planned phase two was set to begin in 2010. But like everything in science and in government, it always takes longer than you think. And actually, the data from phase two is just being released probably in September. And we'll talk about what phase two constitutes in the next couple slides. So what is in vitro high throughput screening and how is it being applied in toxicology? It's essentially taking a number of these technologies that were primarily developed in the pharmaceutical industry and incorporating them in a slightly different way to, instead of screening for therapeutics and potential activity of those therapeutics, trying to screen these chemicals for what biological targets these chemicals may interact with, except doing it with respect to potential toxicological effects instead of pharmacological effects. So it's two sides of the same coin. And they primarily use robotics to screen compounds, just like they do in pharma, in multi-well formats. And their screening assays constitute either cell-free biochemical assays of purified protein, or cell-based assays. So you have a cell-based phenotypic assay, either morphological changes or things like a cellular reporter assay, trying to look for particular activity in a particular pathway, or phenotype readout. And there's also an effort within both of these programs to use lower order organisms such as zebrafish in order to screen these libraries as well. But keep in mind that in these toxicological libraries, as you'll see in the next slide, that they're not the million compound libraries that are being screened by pharma. These are primarily limited to about 10,000 or so chemicals that are considered to be the universe of chemical space that needs to be evaluated for toxicity. So it's a much different magnitude in terms of the number of chemicals that are being screened. But unlike pharma, that wants to screen a specific target, right, to find a therapeutic for a specific slice of biology, in toxicology, you don't necessarily know what effect you're going to be chasing. And so in an ideal world, you're screening these limited number of chemicals across a much broader suite of biological space. So it's a different problem in pharmaceuticals, where it's a large chemical space looking for a very narrow biological space, and instead you're looking for a much broader chemical space, a much more narrow chemical space and across a much broader biological space. But in order to be able to attack this sort of problem right away and hit the ground running, many of these initial toxicology efforts began to use these pharmaceutical assays that were developed for a different purpose. So essentially repurposing these pharmaceutical assays to then screen these chemicals across available repurposed pharmaceutical assays to look for biological activity. And typically, unlike pharma that may screen at 10 micromolar, all of these efforts, both within Tox21 and Toxcast, typically screen their compounds in multi-concentration format, trying to develop a dose-response evaluation of these chemicals in each of these assays, right? And it's primarily being used, and we'll get into this later, as a broad survey of potential biochemical and cellular targets for chemicals. What chemical, if you have chemical X, what is it likely to be able to hit in a particular population, predominantly trying to look for potential human health effects. Is it hitting the estrogen receptor at a very low potency, for example? And what effects do we have to worry from potential endocrine disruption? Or is it particularly cytotoxic to liver? Is it particularly cytotoxic to potential zebrafish reproductive type assessments? So there's a whole range of different types of assays and endpoints that they're looking at. So the chemicals in these libraries are summarized in this particular slide. In Intoxcast Phase 1, we spoke about Toxcast being a phased effort. Well, Intoxcast Phase 1 was broken out into phased efforts for a particular purpose. In Intoxcast Phase 1, they predominantly focused on a fairly small library of chemicals, predominantly pesticides and high production volume chemicals that had a very data-rich database associated with them. So predominantly those that had a very significant in vivo set of animal studies that then could be benchmarked against these in vitro high throughput screening assays. And like it or not, as we saw in one of those slides with the bar graphs, right, there isn't exactly a very rich database of animal studies in order to tap into. And predominantly these data-rich chemicals are things that have regulatory demands that animal studies have to be performed. And these are predominantly these pesticides as well as high production volume chemicals. Phase 2 library is much more diverse, so there's a less emphasis on trying to collate chemicals that have a rich data set, and they are expanding out in terms of the breadth of their chemicals, and not necessarily all of those chemicals are data rich. And so the idea was, at least initially, that you try to identify what potential assays or endpoints you should be concerned about within Phase 1, and then try to validate those within a Phase 2 type of a screening effort. Tox 21 library are much more diverse chemically, but as you'll see on the next slide, they focus on a much narrower suite of assays, predominantly for cost reasons, right? Tox 21 phase library consisted of about 3,000 chemicals, and phase 2 was closer to 8,000 chemicals, trying to look more broadly across chemical space. Compounds that are particularly relevant to food and nutrition is that this Tox 21 libraries contain about 160 GRAS substances. So you'll be able to, you will be able to evaluate the potential activity of these GRAS substances across the assays I'm going to show you in the next slide. So do some of these GRAS substances activate certain cellular receptors or kinases or other sorts of activities in these particular in vitro assays? And there's about 905 FDA EFS substances. I think I'm not as an expert on that particular database, but it's essentially everything added to food in the United States. It's a database that the FDA collates for everything that's potentially added to food in the U.S. And that this database, or this set of chemicals, the 8,000, contains 905 of those particular compounds. And they're screened across a broad range of assays. In Toxcast assays, they contain about 600 to 700 in vitro assays across 10 different platforms. You can see kind of the breakdown of those platforms. You can read that on the left-hand side. But essentially, they are, as I said before, a mix of biochemical assays, purified enzymes, as well as cell-based assays like reporter assays, as well as morphological type imaging assays, looking for the activity of these chemicals across multiple different cell types and different platforms as well. Toxcast assays cover about 327 gene targets. So it gives you a feeling for the amount of biological space that's predominantly covered by these assays. And at least one gene out of about half of the KEGG pathways, if you want to look at biological space in terms of a pathway context instead of a gene context. And the Toxcast assays are primarily repurposed pharmaceutical assays. And because the center within EPA is a virtual center, all of those assays are essentially outsourced to commercial companies. So they don't do any of the experiments within the EPA on those particular screening assays. It's all outsourced. Tox21 is a little different. Remember, they had the 8,000 or so chemicals, but they have a much narrower biological focus. So instead of the 600 to 700 assays that were run in the Intoxcast effort, they're screening those 8,000 chemicals on about 50 to 100 of these in vitro assays. So, and these are primarily cell-based assays, reporter assays that are run at the NCGC. And that is stated down there. So, oh, and I will point out that, come on, go back up. Oh, well, it's dragging. That the estimate for the amount of cost that's being put into these EPA efforts is, at least the reported efforts, about $20,000 per chemical to be able to run all those Toxcast 600 to 700 assays across all those, each chemical costs about $20,000. And they have these two efforts. The Tox21 and Toxcast also has different sort of approaches to how these data is being released. And this is particularly relevant to you guys, I imagine, with the GRAS substances and number of components in foods, of how this data is going to be made public. I think Joe touched on this in terms of how this is going to be made public and how this is going to be discussed within both the lay community as well as NGO community and the scientific community as well. Because all this data is being released in a public domain in order for it to be analyzed by anyone, right? In Tox21, their approach is to actually release this data primarily through online repositories, so the PubChem, essentially the NCBI's version of how they're releasing data from the genome. They have a repository in NCBI as well for chemical activity data, and it's called PubChem. And so all the NCGC assays across all these chemicals are being dumped. I wouldn't necessarily call it in real time like the genome effort, but in relative real time is being dumped into this public repository, and so people can look at their activity across all these assays when they are released for these particular assays and chemicals. And typically, there are fewer analysis-related publications related to Tox21. Toxcast is taking a slightly different approach in that they've been releasing data through predominantly peer-reviewed publication. So when they perform a suite of assays, they will then, instead of dumping it into the public domain in real time like Tox21, they will wait, collate that data, analyze it, and do a minimal amount of interpretation and dump it into the public domain associated with a publication. And then once that publication gets accepted, then it's released on their website for people to download and analyze. So it's a slightly different approach. And they have different analysis-related publications, many analysis-related publications. So I'm going to touch on two slides here of highlight what are the potential limitations of the Toxcast in vitro high-throughput screening data and what we've kind of been pushing in our efforts to try to, how this data could actually be used and applied. I'm just going to touch on two of the issues in the interest of time of what this data is actually telling us so far. And some of you may have a suspicion or a bias on what you think it could be used for already, but at least this is our analysis of what we are beginning to look at this data and what it's beginning to tell us. So as I mentioned before, Toxcast had a number of in their phase 1 effort, had about 309 chemicals that were very data rich. And they had associated with them a large number of an animal database associated with them. So we took that 309 chemicals and the animal data associated with it and analyzed the 600 in vitro high throughput screening assays and looked to see from a statistical perspective, essentially using machine learning algorithms in collaboration with the statistical software company SAS, and essentially did a very broad-based effort. So instead of saying, okay, so it's essentially when you take and do a very broad data mining effort, so this is the effort of big data, right? Where you take if you say something isn't necessarily predictive, you want to be able to say that using not just one machine learning or statistical approach, but in order to definitively say that this data isn't or is predictive, you want to be able to evaluate that in a very comprehensive way. All right? And so we partnered with SAS, which has a suite of different software tools that you can actually apply in a very big data way to analyze how predictive is something across multiple different machine learning algorithms, multiple different ways to select which assays are the best predictors of a particular response in a way that leaves very little doubt that you left no stone unturned. So essentially what we did is we partnered with SAS and looked at the predictivity of those 600 in vitro assays to predict in vivo responses in animals. And we benchmarked that against how predictive just chemical structure was to predicting in vivo responses. Because we believed that if these in vitro assays were no more predictive than the chemical structure, why spend the $20,000 per chemical to run these assays when you have all the information for free with the chemical structure, right? And so this was our ability to evaluate these chemicals. And those in vivo endpoints are just summarized here. And essentially they scan the gamut from all the different animal studies you may run on these different pesticides. And they're highlighted in a different way. There are some chemicals that have a D designation. Those are developmental type studies, developmental type studies that are run for teratogenicity. And you can see the next underscore rat or underscore rabbit. So there's different species involved in some of those developmental studies. There are the studies that are designated by the C. Those are chronic multi-year type bioassays, and they also have different species associated with them. And then you'll see scattered throughout there is an M designation, and those are multi-generational reproductive studies. And you can see the bottom line here is what I want to show is essentially that there are 60 of these in vivo endpoints that scan the gamut of being very specific and very focused on a specific pathology to very broad and not focused on a pathology. So things like a developmental rat maternal toxicity that can include everything from very frank or pathological effects to changes in weight gain. It can be very gross. All right. Whereas this particular study that actually was the most predictive was a chronic rat cholinesterase inhibition. So essentially very specific, meaning inhibition of a specific enzyme. So it runs the gamut to being very granular to being very specific. Right? And on the top, on each of these y-axes, are what's called balanced accuracy. It's your ability to predict that particular response using, on the top, these in vitro assays, or on the bottom, chemical structure. And why balanced accuracy? Well, balanced accuracy reflects both sensitivity and specificity. And the reason you can't use regular accuracy is that many of these endpoints have an imbalance in the number of positive and negative chemicals. So if you have a particular imbalance, let's say you have a particular endpoint that only has 10 chemicals that gives you a particular response. And you have 100 chemicals total, so 90 of them are negative, right? And if you have a really crappy model that predicts everything as negative, you could still have an accuracy of 90%. Right? But your sensitivity is practically zero, or is zero, and your specificity is 100%. And so a regular accuracy wouldn't necessarily reflect how poor your model behaves. Right? Well, balanced accuracy is essentially the average between sensitivity and specificity. And in this particular case, balanced accuracy that I alluded to would be 50%, which would be as good as flipping a coin, and would essentially reflect more of the behavior of that particular analysis and that particular model. And the ideal balanced accuracy is 1, and 0.5 is, as I mentioned, flipping a coin. On the top, as I mentioned, in vitro assays, and the bottom is chemical structure. And across these particular box and whisker plots is a range of performance across all those 84 statistical models. So how robustly can you predict a particular in vivo response from these in vitro assays? And what it's essentially telling you, if you haven't figured it out already, is that these particular 600 particular in vitro assays aren't very good at predicting in vivo responses. And in fact, they're no better than chemical structures predicting in vivo responses to a chemical than chemical structure, right? So essentially, you cannot predict, at least using the current set of data that we've particularly mined, you can't predict in vivo animal responses from these current set of in vitro high-throughput screening assays. You can't predict hazard. But one thing that you can do, another analysis that we did, and a little nuance to that particular analysis, is can you run a slightly different analysis that looks at, okay, I may not be able to predict a particular response, but can I identify what potential mode of action that particular chemical may act by? And in this, we ran I won't go into details, but it ran a particular 2x2 table, which is an epidemiological analysis looking for an odds ratio. So if you have a particular response, particular endpoint, is it enriched for particular cellular targets, chemicals that hit a particular cellular target? And just as an example, we looked for cholinesterase inhibition in the rat. Do they hit the cholinesterase biochemical assays? And are you essentially getting a significant odds ratio? Even though I can't predict with high certainty a particular chemical causes cholinesterase inhibition, I can say with a reasonable odds ratio that that particular response, a chemical inhibition of that particular enzyme can lead to a particular response or a particular pathology. You're enriched in hitting that pathology, right? So it's an odds ratio approach instead of a predictive accuracy approach, which is a slight nuance, but it's actually providing you a more biological perspective on what these potential assays can do. If a particular assay, for example, hits a particular receptor, I can't say with certainty that ultimately that receptor is going to ultimately give you tumors after a 2-year bioassay. There are many other biological factors at play and ultimately determine whether you have tumors. But I can tell you that if you hit a particular receptor, you're at an odds ratio of 10-fold greater risk of getting tumors 10 years down the road just because you're hitting that particular receptor. There are other factors at play than just hitting that receptor and ultimately these more complex biological responses. So some of the current limitations of these Toxcast and Tox21's approach that have really emerged since these efforts began. And one of them is that the current assays, obviously, for those that are biologists in the group, that they may not capture all the biochemical cellular responses in the in vivo tissues with adequate fidelity. It's very well known that these particular assays aren't necessarily have metabolic competence. And so and given that a number of these chemicals require metabolic activity in order to cause toxicity, that's a significant limitation of these assays. And it's one that's being worked on. Right? And they also don't necessarily capture the cell-cell interactions that are important for many of these toxicological responses. Two, the current suite of assays may not reliably capture context-specific outcomes. Right? And that's if you have, for example, NF-kappa B, it can be pro-apoptotic or anti-apoptotic depending on how it's activated. Right? And if you have a cellular assay in a Petri dish, it may not capture the context, the proper appropriate context of how you're activating that particular pathway. And the current set of assays, although you have 600, 700 assays, it may not capture a sufficient number of biological space in order to adequately predict those in vivo responses. And all these assays are run in human cells and in predominantly human proteins. And what they're trying to do is actually predict responses in rat using high-throughput screening assays that are primarily human-based. And so there's a discordance between the species they're trying to predict and ultimately the species of the assays that they're actually screening. Ultimately, the goal is to predict human responses, but in order to benchmark this of what we've already what we've already done for the last 30, 40 years in these animal studies, if we're going to benchmark against that, you also have to understand that particular context and that limitation. And the current analysis does not capture assay efficacy, or is 300 chemicals really enough in order to fully evaluate whether these particular assays are predictive or not. So why is an example of why context is important from a dietary perspective, and this is probably more interest to you guys, is that one of the things as we were been evaluating these particular assays and what they could and couldn't tell us, this study, as I was doing some research and before a talk that I was supposed to give at one of these in vitro high-throughput screening conferences, this set of articles actually surfaced because it provides, at least for me, a good example of why context is important, right? So as I mentioned before, a big discussion in the tox community are around these toxicity pathways. If I activate P53, is that a bad thing from a chemical, right? If I particularly activate PPAR alpha or estrogen receptor, is that a bad thing, right? Am I going to, in all cases, right? And so this set of articles actually took 13 men in a retirement home and took blood from those men pre-diet and then put them on a diet of low-fat, high-fiber diet, and it made them exercise, heaven forbid. And from those blood samples, they isolated serum, and then they took those isolated serum pre-diet and post-diet and actually used them in a cell culture assay. Essentially exposed cells in culture, prostate cancer cells in culture, a cell line, to these serum samples pre-diet and post-diet. And lo and behold, from those pre-diet and post-diet samples, those cells actually post-diet underwent a higher rate of apoptosis and actually activated p53 in the diet and exercise group. And in the particular thinking, at least in this new thinking about Toxcast, Tox21, and the switch to in vitro high throughput screening assays, toxicity pathways, right, that this particular set of assay results, if it was a chemical, would actually be raise red flags of saying, hey, if this was a chemical, you may be worried about it because you're activating p53, causing cell death. Right? But in matter of fact, that what's being evaluated in this particular assay, if you didn't know it, is the fact that low-fat, high-fiber diet and exercise actually in this particular context, activating p53 is a good thing, at least. And now I can tell my wife, though, that I shouldn't exercise and I should not eat very healthy, right? Because it might activate p53. But this just highlights the potential context and how these particular assays need to be interpreted in the appropriate context. So finally, a project that, as it was alluded to by Joe, that ILSI is contributing to, that we're trying to provide at least a little bit of this context to these Toxcast high-throughput screening assays. And essentially what we're going to be doing, instead of screening chemicals, is screening about 30 fruit and vegetable juices in concentration response format across the same set of Toxcast and Tox21 assays. So these will be done at 6 to 8 concentrations with a maximum of 10% juice. Screening a subset of cell-based and biological assays. These are the same assays that are being used by Toxcast and Tox21. So essentially we're going to contract with the same people that Toxcast is going to be contracting with. And we're going to follow a protocol that's already been developed for screening fruit and vegetable extracts across these particular assays. We can talk a little bit more about the details, but essentially they screened 30 fruit and vegetable extracts. In their particular case, they did it for genotoxicity. They did it for in their limb bud formation assay, looking for potential terata from these particular fruit and vegetable extracts, as well as for estrogen activity. And they showed in this particular paper that a number of fruits and vegetable juices actually had significant, obviously, genotoxicity. They actually had significant estrogenic activity. You can see that, as you may expect, things like snow peas and whereas my favorite soybeans obviously have estrogenic activity, and in some cases almost as much as estradiol, right? And screening in this particular context. And so it actually provides a context to these particular chemicals, screening in these fruit and vegetable juices. And the Tox21 group is also interested in this project, so we're collaborating them. They're going to also be running these juices in their assays at the NCGC. And in theory, the way that, the juices will be obtained from organic fruits and vegetables. Not that necessarily organic means that they're pesticide-free, but there's been also a study in the literature suggesting that activity in these in vitro assays is more associated with the pesticide residues than the natural chemicals in the juice or in the vegetables. Not that I believe that study, but in order to counter those particular criticisms, ILSI is contributing the fact that they're going to be screening these particular juices for pesticide residues prior to screening. And with that, I'm out of time, and so I'll acknowledge everybody in my group and my collaborators who've collaborated with me and supported me, and my funding from the ACC, LRI, and ILSI, and I'll try to answer any questions that I can.